Aloha, everyone. This video is for January 25th and 26th, so starting week 4. Today, we will start with a punnet bell ringer just to keep practicing our punnet squares. Then we're going to move on. We kind of have to backtrack to move forward, but we're going to talk about cell division this week. We're going to start with a mitosis Edpuzzle. And then after the Edpuzzle, we're going to do a video recap worksheet. It's going to be about the same video. It's just an additional assignment so that we can practice drawing the stages of mitosis. So the first one is the Punnett Square bell ringer. So we're going to go to Canvas and modules like usual. And we're going to be under week 4 here. So again, this page tells you everything about this week. We'll even include this week's lesson agendas. But we're going to start down here underneath that with punnet bell work and today's date. Okay, so it's very similar to the last one. I'm going to give you a problem and then you have some questions. So in this problem, it says, let's say that in seals, the gene for length of the whiskers has 2 alleles. The dominant allele, shown as the big W, codes for long whiskers, and the recessive allele, coded with the small w, codes for short whiskers. What is the percentage of offspring that would be expected to have short whiskers from the cross of 2 long whiskered seals, one that is homozygous dominant and one that is heterozygous? So very similar to the last time we did this, the first few questions are just to understand, or just to make sure that you understand what information we need and where to find them in the problem. Number 1 asks, what is the genotype of a seal that is homozygous dominant for long whiskers? Number 2 asks, what is the genotype of a seal that is heterozygous for long whiskers? Number 3 asks, what would the genotype be of a seal that has short whiskers? So number 1 is one parent, number 2 is another parent, and number 3 is the genotype of what we're looking for, right? So then on a separate scrap sheet of paper, create that Punnett square. Now, I highly suggest writing this down. Don't do it in your head because that's where mistakes get made sometimes. So write it down. You don't have to show me what you did. You'll show me in the answers, but please make sure that you do it. So for number 4, you're going to list out the genotypes and the phenotypes of the 4 babies. So this is an example. Do not use this example if it's not the correct answer. So for example, if I had these 4 babies, this is a genotype and it's phenotype. Genotype, phenotype, genotype, phenotype, genotype, phenotype. Make sure I have 4 babies and you tell me what those 4 babies' genotypes are and those phenotypes. So use whatever you get from your Punnett square, not my example. And we're working with whiskers, not dimples, and W's, not B's. So make sure you're working along with what is in our problem. The last question is the question that you're answering in this problem. What is the percentage of offspring that you would expect to have short whiskers? Okay, so for 4 and 5, you should be able to get after you create your Punnett square. Now you could just click submit assignment here and a text box will pop up. And then all you do is answer questions 1, 2, 3, 4, and 5 and click submit. If you find yourself confused as to what genotypes and phenotypes are, remember that I have notes all over Canvas about this. So here's the Mendelian genetics vocabulary slides. I'm going to go back to modules to show you where I found that. So here's this unit we've been working on. Mendelian genetics vocabulary, if you need recap on your vocab. Punnett squares, if you need recap on how they work. And of course, any of the in case you missed it videos will also have that information. But genetics terminology, you need to make sure that you understand the vocab This whole thing talks about the vocab that we're using. So if you get confused as to what genotypes are, if you are confused as to what homozygous and heterozygous genotypes are, this is a great resource. Same with if you forget what phenotypes are. All right, when you're done, turn that in. And that's it. When you're done with the bell work, we're going to move on to the Ed puzzle. So it'll be down here right underneath where you went for the bell work today. It's called Cell Cycle and Mitosis Ed puzzle. So you'll scroll down and go to whichever language you want to do the Ed puzzle in. Click on your class period. Okay, please make sure that you click on your correct class period. Watch the video carefully and answer each question. And then when you're done, it'll save automatically, but unfortunately, Edpuzzle does not tell me when you all do the Edpuzzle. So I want you to go to Submit Assignment, just like the bell work. And in this text entry box, I just want you to tell me when you're done that you are done. And you'll submit it that way. And then that's it. We've done Ed puzzles before, but if you're new to them because you're new to this class, or you're just not sure how they work, let me know, and I'll be happy to help. Lastly is the mitosis video recap. So we just watched the Ed puzzle over mitosis, and if you watched it in English, it was Amoeba Sisters. In this assignment, you're going to do the recap worksheet that goes along with the video. So you will actually work on this in ICYMI because you're going to draw on it. So you're going to follow these directions and use ICYMI. If you have forgotten how to use ICYMI, here are some instructions for that, for getting this document uploaded there. This is what the worksheet will look like. Okay, there are 6 questions that the video talked through, so you might be able to remember some from the first time you watched it, like not too long ago, but of course you can rewatch it if you need to. The part I really wanted to focus on was sketching the stages of mitosis. So you learned about PMAT, prophase, metaphase, anaphase, and telophase. So in these boxes, I want you to draw what the cell looks like when it's in that stage. And I want you to include labeling chromosomes, spindles, and the nucleus if it's applicable. So you do that for all 4 of those stages. And ICYMI gives you the option of drawing, so that's why I had you use ICYMI. To go back to the video, you don't have to keep watching the Ed puzzle version because it stops every so often for those questions. So I have linked here the YouTube version so that you can pause it, rewind it, fast forward, move through this video as much as you need to get this done. When you're done, you're going to download it from ICYMI, remember, with annotations, and you're just going to submit it here through File Upload. If you have any questions on any of the assignments we did today, please let me know. Otherwise, have a great rest of your day.